So in our previous class we have dealt with Phycology-1, where we have learnt diverse habitats of algae, habits of algae, and then we have exposed to vegetative characteristics like pigmentation, cell wall composition, different types of algae from unicellular to filamentous forms. And today we are going to start with the reproductive part of algae. Okay, types of reproduction in algae. Algae will have three types of reproduction. We have vegetative reproduction, asexual reproduction, and sexual reproduction. First, look into the vegetative reproduction. Vegetative reproduction occurs in algae only during favorable conditions, not in unfavorable conditions. Only during favorable conditions, vegetative reproduction occurs in algae, and mechanism here is this vegetative reproduction is always uniparental. It is uniparental and it occurs only in the favorable conditions. A portion of thallus, this gets separated and gives rise to the new individual. This gives rise to new individual. Based on Thallus organization, we have three kinds of vegetative reproduction. Okay, we have three kinds of vegetative reproduction which are not shown. But here you see what are the three common modes of vegetative reproduction. We have first is what? Fragmentation, second will be fission, and third is budding. Okay, fragmentation, fission and budding. Fragmentation, where from a single parent, okay, for example if you consider a filamentous algae, small fragment of this detaching from the parental thallus, the small fragment will completely give rise to a new thallus or new individual. Fine, right. Next, fission. Fission is common. Fission is common in unicellular algae. Binary fission is common in unicellular algae, where the cell will divide mitotically into two cells. Single cell will divide mitotically. Single cell will divide mitotically into two cells. Next, budding. A new individual will develop from small outgrowths or buds. They will develop small outgrowths or buds of the parent, which are less common in algae. So what is the most common mode of vegetative reproduction? Most common mode in multicellular or filamentous algae is fragmentation. In unicellular algae, most common mode of vegetative reproduction is fission. I hope these three are clear to you, and then we will move on to cell division and autocolony formation. How does this happen in the algae? Common in unicellular, motile and non-motile algae. Cell division is most common method to increase the cell number in the algae, wherein here sequential karyokinesis is followed by cytokinesis. Okay, that is nuclear separation is followed by cytoplasm separation. For example, here you see Crocococcus and Gliocaps are the examples for what? Karyokinesis followed by cytokinesis. In desmites and diatoms, each cell will have two semicells like this. A single cell will have two semicells like this. This is observed. This is observed in Cosmarium and Pinularia. Okay, this is due to partial cytokinesis. Karyokinesis followed by partial cytokinesis. Okay, till here I hope it is clear right now. And then you see peridium or dinophycy. Peridium or dinophycy. Parental wall dissolves and new walls form for daughter cells. The parental wall will dissolve and new wall is formed for daughter cells like this. New wall formation occurs for daughter cells. This mode of division is happening in where? Peridium, that is dinophycy. Next, euglenophycy. In euglenophycy, longitudinal division will divide it into two valves. Okay, longitudinal division will divide it into two valves. Fine, right? So, karyokinesis followed by cytokinesis in Crocococcus and Gliocapsa. Desmites and diatoms, two semicells are present inside an individual cell. In Peridium or Dinophysi, what happens? Parental wall will dissolve and new wall will form in the daughter cells. Then in Euglenophysi, longitudinal division will divide it into two equal valves. Fine, right? In binary fission. Next is autocolony formation. In autocolony formation, this is seen in cenobic algae. Okay, autocolony formation, this is observed in cenobic algae like Pediastrum and Hydrodictyon. Fine, right? Autocolony formation is seen in cenobic algae like Pediastrum and Hydrodictyon. Here a miniature colony, a small colony is produced by a parental colony. From parental colony, a small colony is produced. From parental colony, small colonies are produced. Fine, right? These small colonies are addressed as miniature colonies as they are formed directly from the parental colonial cell. This is called as autocolony also. Okay, and then this is formed by repeated bipartitions of cenobial cells. Okay, repeated bipartitions of cenobial cells will result in this small miniature colony formation. In volvox, autocolony develops from specialized cell called as gonidium. Remember this, very important statement. In volvox, what is the autocolony that develops from the specialized cell? What is the name of that specialized cell? Gonidium is the name of that specialized cell from which the autocolony is formed. Next is fragmentation. Fragmentation is most common in filamentous green algae. Okay, fragmentation is most common in filamentous green algae. For example, Spirogyra, Zygnema, Eulothrix and Cladophora. Okay, filamentous green algae is most common in this. The thallus will break into small fragments, each of which will grow into a new plant. Each fragment will grow into a new plant. Fine, right? So, fragmentation is most common in filamentous green algae, from which each small fragment will grow into a new plant. Clear till here? Any doubt anywhere, ma? And then every point is important for your exam. Fine, right? This table, if you want, take a screenshot of this, but the examples of each type of vegetative reproduction is important for you, ma. Each type is important. Here we go with first, Hormogonia. What are these structures? Short fragments of trichomes. These are separated by necridia. Okay, where the cells which are older, they will be dying due to autophagy. Okay, for survival and propagation due to necrosis. In the filamentous algae, the dead cells due to necrosis whichever are formed, they will form some trichome like structures which are known as what? Hormogonia. This, when dissociated from the parental filamentous thalli, the small fragments will give rise to new individuals. The structures Hormogonia, where are they found? They are found in nostoc, oscillatoria, which are blue-green algae, or cyanobacteria. Fine, right? Which belongs to what? Primitive algae, cyanophysian members. Next is propagules. Second one is propagules. These are 1 to 3 celled adventitious branches from thallus. They develop into new plants. Okay, they are 1 to 3 celled adventitious branches. They are from thallus which develop into new plants. They are found in where? Spacilaria, which belongs to which class? Phaeophysi. They belongs to Spacilaria, which belongs to Phaeophysian class. Next is Gyma. Okay, Gyma, these are microscopic multicellular structures. They detach from the new individual. Where do you see Gyma? Gyma you usually see in the liverworts. Okay, Gyma structures are usually observed in the liverworts like Mercantia, but then they are also present in few advanced algae. Okay, these are small microscopic structures. Okay, they will detach and they will give rise to new algae or new gametophytic algae. Best example that you can see, where do you see Gimma? Gimma you will see in the advanced class, that is Rhodophyceae, and the algae you will see here is Heldenbrandia rivularis, rivularis, which is a Rhodophycean member. In this you can see Gimma. Okay, and then bulbils. Bulbils are multicellular bud-like outgrowths. These are multicellular bud-like outgrowths which grow on the thallus. They will develop into a new individual. Where do you see bulbils? In Chara, which belongs to what? Characeae, Charophyceae. Okay, and then if you see adventitious embryos. Adventitious embryos, these are multicellular embryos. They are formed on thallus. They develop independently. They are formed on thallus and they develop independently. Example you see is fucus, which belongs to what class? Phaeophycean class. Then achinates. Achinates are thick-walled resting cells. These are thick-walled resting cells with reserve food material. They will germinate under favorable conditions only. They are found in Cylindrospermum, Gliotricha, Zygnema, Pithophora and Stygioclonium. Okay, these are the examples where do you observe achinates. Don't get confused between these two structures. One is Hormogonia and other is achinates. Achinates are thick-walled resting cells and Hormogonia are small trichomes which are formed due to necrosis or necredia. Okay, and then is statospores. What are statospores? Statospores are thick-walled spores. These are smooth or ornamented. They can be smooth or ornamented. They are thick-walled spores. They are found in diatoms, Vaccaria and Geminata. Okay, Acromonas dinobryon. These are the examples where do you see statospores. Statospores are thick-walled spores. They are produced due to unfavorable. Next is hypnospores. Hypnospores are also same like statospores, which are thick-walled spores. Okay, like this, they are thick-walled spores, but both daughter cells they will develop thick walls. Both the daughter cells will develop thick walls. They will resist the stress. Example here you can see Ulothrix fimbriata. This is the example, Ulothrix fimbriata. Next is resting cysts. Resting cysts. Thick-walled cysts, they are formed during unfavorable conditions. These are thick-walled cysts. They are formed during unfavorable conditions. Example is Cylindricum, Astabularia and Nemealion. These are the examples where you can see resting cysts. Each and every structure is important which is facilitating the vegetative mode of reproduction. Clear? You can take a screenshot of this one. Fine? And now asexual reproduction. Okay, asexual reproduction. This is uniparental. Fine, right? Asexual reproduction is uniparental. Vegetative reproduction and asexual reproduction. What is the primary difference? Vegetative reproduction is directly from the thallus of the algae. Okay, any part of the thallus will detach from the parent and that gives rise to a new individual. That is what? Vegetative reproduction. Whereas asexual reproduction, if you see, that is purely by spores. Both type of spores you will come across, the exospores and endospores. And same like that of vegetative reproduction, asexual reproduction is also observed during favorable conditions only. Okay, asexual reproduction is also observed during favorable conditions and asexual reproduction is also uniparental. Okay, single parent will be there. This involves specialized cells or organs without fusion with other cells. There will not be any fusion with other cells. Here you see exospores and endospores. Two types of spores are produced, exospores and endospores. Exospores are formed in non-filamentous cyanophycean members, which are primitive algal members. Primitive algal members. Fine, right? Exospores, they are formed in non-filamentous cyanophyceae. The distal end of protoplasm, this will cut off successively. The distal end of the protoplasm will cut off successively, and the cells are released outside the spores. Cells will be, right? How exospores are being released? The distal end of the protoplasm, they will cut off successively and cells will be released outside as spores. Example here you see chemosiphon. Example is chemosiphon. Next is endospores. Endospores is observed in cyanophyceae. Cyanophycean endospores are also observed. Example is dermocarpa. Dermocarpa is the example. What is the formation process? The entire content of the cell is divided into small and non-motile elements. Fine, right? The entire content of the cell is divided into small and non-motile elements. The division occurs either successively or simultaneously. Okay, division here occurs successively or simultaneously. What is the release? Mode of release of this endospores. The mother cell wall will degenerate, allowing the spores to be liberated. Okay, the cell wall of the mother cell will degenerate and the endospores will be released. How exospores are released out of the cell? Exospores will be released from the distal end of the protoplasm. Once the spore is cut off, it is cut off successively, and this is released from distal end. Whereas endospores, they will be released once the cell wall is degenerated from the parent plant. Clear with two types of spores of asexual reproduction? I hope this is clear. And then types of asexual spores in algae, like how we have looked into the structures which facilitate the vegetative reproduction. Now we have to go through the spores which aid the asexual reproduction in algae. This table is very important, ma. Please take a screenshot. This is highly useful for you, and revise it multiple. You can easily answer the question which is coming out on concept to be addressed. So here you see this monospores. Monospores will be having uninucleate protoplasm that transforms into a single spore in a monosporangium. Fine, right? Monospores are having uninucleate protoplasm and they will transform into a single spore in a monosporangium. Examples if you see Rhodophyceae, Phaeophyceae, Conifera, Alba and Porphyta. These are the examples for monospores. Next, bispores. Two spores present per sporangium. Okay, per sporangium, two spores are present. Examples if you see red algae, Calithamnion, Lithophyllum, these are the examples where bisporic conditions are observed. Paraspores. What are these paraspores? They are clustered masses of spores in the sporangium. Many spores. Okay. Is the voice clear now? Is the voice clear now? Clustered mass of spores is known as paraspores. For example, Ceramium striaticum, which is seen in Rhodophycean members. Is the voice clear now, ma? Aplanospores. Aplanospores are what type of spores? Non-motile spores. They are formed under dry condition and they are dispersed by wind itself. What is the mode of aplanospores dispersal? Wind, as they are non-motile spores. Where do you observe aplanospores? Criboxia and Botrydium. Okay, these are aplanospores. Next is autospores. Autospores are formed by segmentation of the parent cell. By partition of the parent cell. Each spore will resemble the parent and they have distinct wall. Each spore will resemble the parent and they will be having a distinct wall. Examples you see, Schenidesmus, Periaster and Chlorella. These are the examples of autospores. And then is zoospores. What are zoospores? They are most common uni, bi, quadri or multiflagellated spores. They are from zoosporangium. Okay, they are from zoosporangium. Examples you see, Gonidium, Chlamydomonas, Eulothrix. These are the examples for zoospores. Most important, zoospores are usually biflagellated. Of course, it can be uni, bi, quadri or multiflagellated, but mostly it is biflagellated. Mostly it is biflagellated. Clear? Next is Stephanocontic zoospores. These are spherical zoospores with crown of flagella below the tip. Okay, so here you can see spherical zoospores with crown of flagella at the tip. This type of zoospores, Stephanocontic zoospores, are observed again in Erogonium, Darbacea and Bryopsida neglectica. Fine. And then you see schizospores. These are multinucleated spores. Okay. Multinucleated spores, which are motile spores with two flagella opposite the nucleus. Opposite to the nucleus, they will be having two flagella. Example is Vocaria xanthophyci. Here what is that you have to remember most? The type of spores and the examples in which the spore type is present. Fine. Description though you can easily address from the name of the asexual spore. Fine, right? Just go through this. If you want, take a screenshot. Fine, right? And then sexual reproduction. Here we go with this. This occurs usually before the onset of unfavorable conditions. Sexual reproduction starts before the onset of unfavorable conditions. This is unlike vegetative reproduction or asexual reproduction, which occurs only during favorable conditions. This is absent in Cynophyceae. Okay, sexual reproduction is absent in Cynophyceae. In Xanthophyceae, only Vocaria exhibit sexual reproduction. Only Vocaria exhibit sexual reproduction in Xanthophyceae. This involves fusion of two specialized reproductive cells. Fusion of two specialized reproductive cells. What are the stages of sexual reproduction? Three stages. One is plasmogamy, which is fusion of cytoplasm. Second is karyogamy, which is the fusion of nuclei, and third is meiosis. This occurs early or late meiosis. Fine, right? Now, you see, we have three types of sexual reproduction. One is isogamy, where two gametes are of same morphology and physiology, whereas anisogamy, the gametes differ either in their structure, that is morphology, or in the physiology. Third is oogamy. Okay, oogamy, wherein the female gamete will be large and non-motile, male gamete will be small and motile. As such, we have three types. What are they, ma? Isogamy, anisogamy, and oogamy. First, we will look after isogamy. This is simplest and most primitive type of sexual reproduction. This involves fusion of morphologically identical gametes. They will be physiologically identical as well. Here we go, gametes of the same individual. If the gametes are from the same individual, then you will address that thallus as homothallic. Single thallus will give rise to two gametes. male and female gametes arise from single thallus. If two different thalli are producing two different gametes, then it is addressed as heterothallic. Two different thalli are producing different thallus, then it is what? Heterothallic. Examples you see here, Chlamydomonas and Ectocarpus. These are the examples for what? Isogamous type. Then we will see what are the types of this isogamy. Flagellated gametes. Flagellated similar gametes are observed in Chlamydomonas, which is wide known example for all of us. Non-flagellated gametes or aplanogametes, they will be Spirogyra and Zygnema. These are non-flagellated or aplanogametes. Next is post-fertilization. Zygote will undergo nuclear division, that is karyokinesis. They will form 4 cells. 2 will have positive strain zoospores and 2 will be negative strain zoospores in dioecious species. Fine, right? Post-fertilization, the zygotes will be either positive strain or negative strain. Fine, right? Which will be acting as male gametes and female gametes further. Fine, right? Here what you have to remember more, homothallic and heterothallic species, followed by what are the flagellated, non-flagellated gametes of isogamy. Examples will play crucial role in every slide of this aspect, every slide of this concept. Examples will play crucial role. Fine. And then anisogamy. Anisogamy is fusion of dissimilar gametes. Dissimilarity is either in terms of morphology or in the terms of physiology. Anisogamy, dissimilarity is either in the terms of morphology, morphological difference, or physiological difference. Fine. They are produced in gametangia. Smaller gamete will be male, which is active. Larger gamete is female, which is passive. Fine, right? And then is oogamy, which is advanced sexual reproduction in algae. This is the advanced sexual reproduction in algae. Oogamy is most advanced type of sexual reproduction. In this type, male and female gametes will differ both in size, motility, behavior, shape and structure. Gametes are produced in specialized sex organs like antheridium and oogonium. They are produced in specialized sex organs. Most algae are produced, they will produce a single ovum or egg cell in the oogonium. What is the exception? Fucus and sargassum. They are the only exception. Fine, right? For oogamous mode of reproduction. Clear both anisogamy and oogamous type? Any doubt anywhere, ma? Shall we move on to the next concept? Yeah. Oogamous type of sexual reproduction is mostly or widely observed in rhodophycean members, where male gamete here is non-motile but it is small, which is spermatia. This is known as spermatia. Male gamete is known as spermatia and female sex organ is known as carpogonium. Male is spermatia, female sex organ is carpogonium. Fine, right? Advanced method of sexual reproduction in algal members is observed in rhodophycia only. So here trichogyne structures. These are tubular outgrowths from the female sex organ carpogonium. They will receive the spermatia which are carried across the water currents, and this spermatia will adhere. The spermatia or the main gamete, they will adhere or fuse with the ovum, okay, through this oogonium. Fine, right? And this structure is known as what? Trichogyne, where the male gamete, that is spermatia, will come and fuse with the female gamete. Here zygote formation. Zygote formation. Zygote of the fresh algae will always have a thick wall. This enters a resting stage and then germinates into a new individual. Okay, zygote will germinate into a new individual only during favorable conditions. What is the significance of sexual reproduction? First, it prevents degeneration. it prevents degeneration of the race, which may result from continuous asexual reproduction for an indefinite period. Second is, this introduces genetic variation. Fine, right? Offsprings will differ from the parent due to random gene combinations during gametic fusion. Third is, it produces more offsprings than asexual reproduction. Sexual reproduction will produce more offsprings when compared to asexual reproduction, where natural selection will promote survival of the strong individuals only. This will improve the balance. Last advantage of sexual reproduction is this will aid in evolutionary process, where this contributes to biodiversity and adaptability. Clear till here? This is the significance of sexual reproduction. Fine. And then what is the classification of this? Carolus Linnaeus in the year 1754, Carolus Linnaeus in the year 1754 included algae and lichens under cryptogamia. Okay, he has included algae and lichens in cryptogamia, and there is no detailed classification for this. Later, after Carolus Linnaeus, it is the scientist, that is Walker, in the year 1803. They proposed the first classification system of algae, and they divided algae into coniferous, ulvus and tremalus. Criteria for algal classification. What is the major criteria for algal classification? First, composition of photosynthetic pigments. Second, nature of reserve food material. Third will be chemical composition and physical nature of the cell wall. And then structure, number and orientation of flagella. Structure, number and orientation of flagella, followed by prokaryotic and eukaryotic cell organization. These are the major criteria for algal classification. Of course, most of it we have addressed it in the yesterday's class itself about pigmentation, cell wall composition and storage food material. Widely accepted classification is F.E. Fritsch classification, which is widely accepted classification of algae. This gave most authentic and comprehensive algal classification. This is based on pigmentation, reserve food nature, type, number and insertion of flagella, presence or absence of nucleus. They have published 2 volumes of the work, the structure and reproduction of the algae. Here you see algal classification by F.E. Fritsch. He classified algae into 11 classes. What are those 11 classes, ma? Chlorophyceae, Xanthophyceae, Chrysophyceae, Bacillariophyceae, Cryptophyceae, Dinophyceae, Chloromonidae, Euglenophyceae, Phaeophyceae, Rhodophyceae and Mixophyceae. These are the 11 classes of algae, of which in detail we have to learn about characteristics, type studies, major type studies involved in them, followed by economic importance. Of what algae we have to learn? Chlorophyceae, Phaeophyceae and Rhodophyceae. Cyanophyceae as well, which is the primitive algae, where we have to address on nostoc, Anabina and Oscillatoria. Fine, right? These are the major algae that we have to go through. Now let us see each and every class in detail. Okay, every class in detail, their pigmentation, habitat, reserve food material, reproduction, and what are the further orders involved in that. See, orders of each class is not required for you, but in basic terms you should be aware of what is the reserve food material, thallus type, and what is the pigmentation of each class of algae is important. So here you see, ma, Chlorophyceae. Fine, right? First is Chlorophycean members. Mostly they are of freshwater habitat. Pigmentation is chlorophyll A, B, and they will have carotenoids and xanthophylls. Reserve food material is they are having true starch. Okay, starch is the reserve food material. Thallus type, it is unicellular, multicellular or filamentous type. Reproduction is isogamous, anisogamous or oogamous. 3 types of reproduction is observed in chlorophycean members. And you can see wide diversity of chlorophycean members. You can see unicellular forms, colonial forms, filamentous forms and multicellular forms also. So, all the types are observed in different orders. You can go through that. Next is xanthophyceae, okay, yellow green algae. Here you can see the pigmentation, chlorophyll A, E, especially beta carotene and xanthophylls, which gives it yellow green color, yellow orange color also. So, here the reserve food material is oils and fats. Thallus type is unicellular, motile, simple branched and filamentous types are present. Reproduction is usually asexual and sexual modes of reproduction is observed in xanthophycean members, both asexual and sexual types of reproduction. Next is chrysophyceae. They are both fresh and cold water, okay, chrysophyceae, fresh and cold water type. Pigmentation is phycocrescent. Phycocrescent is the pigmentation. Reserve food material is fat and leucosin is the reserve food material. Thallus type is unicellular, motile or branched filamentous type of thallus. Reproduction is rare sexual reproduction. Fine, right? You will observe rare sexual reproduction. Next is Bacillariophyceae. Okay, habitat usually ubiquitous, marine and freshwater habitat. Pigmentation is diatomine. Reserve food material includes fat and volutin is the reserve food material. Fine, right? Thallus is unicellular or colonial. Reproduction is asexual or sexual mode of reproduction. Clear, ma? Everything is fine till here. And then next set of classes, cryptophyceae. In every class, the highlighted point that you have to observe is the pigmentation, reserve food material, habitat, of course that is common, thallus and reproduction type. Thallus mode also is important. Clear? Yeah. Here you see cryptophyceae, where the pigmentation is red, brown, greenish, yellow pigments, which includes phycoerythrin, carotenes and xanthophylls. Reserve food material is starch and reproduction is both asexual and sexual reproduction. Next is Dinophyceae. Okay, Dinophyceae whose habitat is marine and freshwater. Pigmentation is usually chromatophores, wherein it is dark yellow and brown color pigmentation is observed. Reserve food material is starch and fat. Fine, right? And reproduction is rarely sexual reproduction. Next is Chloromonidinae. Okay, this class will have what type of pigment? Pigmentation is important for each class, as I told you. Xanthophylls are the major pigments and the reserve food material is oil. Thallus type is motile with the longitudinal flagella. Next is Euglenophyceae. Euglenophyceae usually habitat is fresh water. Pigmentation is chlorophyll A, B, carotenoids and xanthophylls. Reserve food material is polysaccharide and paramylon is the reserve food material. Thallus type is motile with flagella. Fine. Clear? 4 types of classes. Furthermore, I hope you have taken a screenshot of this. Yeah. Phaeophyceae. Major pigmentation is fucoxanthin. Reserve food material is laminarin and mannitol. Reproduction is only sexual, which is oogamous type of reproduction. As you move on to the advanced classes, the sexual reproduction will be only oogamous type. Rhodophyceae, pigmentation is phycoerythrin, where the reserve food material is Floridian starch, and mode of reproduction is sexual reproduction again, which is oogamous type of sexual reproduction. And lastly, if you see Mixophyceae or Cynophycean species, Mixophyceae or Cynophyceae. Here you can observe diverse pigmentation, wherein that is chlorophyll A, beta carotene, phycocyanin, phycoerythrin, mixoxanthin, mixoxanthophylls. These are the major pigments that are observed in Mixophyceae or Cyanophyceae. Reserve food material is sugars and Cyanophycean starch is the reserve food material. Mode of reproduction is asexual reproduction only. This is the mode of reproduction. Clear? These are the classes. And then, what are the salient features of Chlorophyceae? Let's begin with Chlorophyceae and we will learn two type studies from here. Of course, first is habitat. Fine. First is habitat, where 90% of them are of freshwater species only. Some of them will be terrestrial and some of them will have brackish water forms. Second will be pigmentation, where the pigments pigments will have chlorophyll A and B. Accessory pigments include siphonoxanthin and siphonin and lutein. These are the accessory pigments or antenna pigments. Next is Thallus Organization. They have high diversity. They are unicellular forms, palmelloid forms, heterotrichous, siphonaceous, follows and complex. All forms of habits we have learnt in the previous class. You can go through the class ones. Next is cell wall composition. Cell wall composition, their cell wall is made up of cellulose. They also contain silanes and mannans. Reserve food material is pyrenoids which are present inside the chloroplast. Next cell wall composition is done. Chloroplast structure will contain pyrenoids, and how many thylakoid groups are present? 3 to 5 thylakoid groups are present in each chloroplast along with the reserve food material structures. Those are pyrenoids. Pyrenoids consists of starch inside it. Next is photosynthetic product. Photosynthetic product will be obviously starch. Starch is the photosynthetic product. Fine. And sexual reproduction is isogamous, anisogamous and oogamous. All the three types of sexual reproduction is observed, and mostly the members will be haploid. Life cycle is, we can see three modes of life cycle. When you consider the diverse examples of Chlorophycean members, you can see three life cycles: haplontic, haplodiplontic and diplontic life cycle. Fine, right? And then haplontic, haplodiplontic and diplohaplontic life cycles are observed. First we will see unicellular green algae, that is chlorella species. What is the occurrence of the chlorella? Here you can see the occurrence of the chlorella. They can be in the fresh water, marine water, and they can also be present as an endosymbiont. Endosymbiont in Paramecium, Hydra and some sponges. They can be as the endosymbionts in this. Next, Chlorella lichena. Chlorella lichena. They form lichen association with fungi. Lichen association is associated along with fungi. Thallus structure. Thallus structure is, it is having unicellular algae and they may appear colonial as well. Cells are spherical, subspherical and ellipsoid. 2 to 10 micrometers in diameter. They are of 2 to 10 micrometers in diameter and they do not have flagella. They contain single parietal chloroplast with pyrenoid. They will have single parietal chloroplast with pyrenoid. Cell wall is made up of cellulose. Cell wall is made up of cellulose and they also contain sporopollenin like structures or substances are present. Fine, right? Cell wall will have cellulose, silanes, mannans are also composed here, and they also have sporopollenin like structures which are seen in pollen grains of higher plants. Sporopollenin like structures are also observed in the cell wall of chlorella members. Done, ma? And then reproduction. Asexual reproduction is through autospore formation. Okay, asexual reproduction is usually through autospore formation. Each mother cell, single mother cell will have 4 autospores. Okay, each mother cell will have 4 autospores, miniatures of the parent cell. Fine, and there is no sexual reproduction. No zoospores or gametes are formed. There is no sexual reproduction. Economic importance of chlorella, if you see, They are highly of nutritional importance. 50% protein, 20% carbon dioxide and 20% of lipids are present, and they contains all essential amino acids are present, and they are rich in vitamins like vitamin A, riboflavin, biotin and folic acid. They contain minerals like iron and calcium. These are the minerals that are present over here. Next, they produce antibiotics called as chlorellin. This is the antibiotic which is present. Fast growth rate is seen. Oxygen producer, they are used, this is the best oxygen producer which is used by spacecrafts to absorb carbon dioxide, and they are used in sewage treatment in oxidation ponds. Dried biomass is used as single cell protein. Okay, dried biomass is used as SCP, that is single cell protein. Next, volvox. Volvox is the freshwater planktonic algae. They are commonly observed or found in stagnant waters. They are found in stagnant waters like pools and ponds during spring and rainy season. Colonies appear as green pinhead structures. The colonies, green algal colonies, volvox colonies, observe as green pinhead like structures. They are visible to the naked eye in over the water surface. Fine. Now, can you see the slide? Might be there is some network issue over there. Here it is clear, ma. Yeah. Thallus structure, colony or cenobium is seen is spherical and it consists of 500 to 50,000 cells. Okay, they have 500 to 15,000 cells, including some vegetative cells and gonidia. They are held together by gelatinous sheet in the form of a colony. They are held together by gelatinous sheet. Next is vegetative cells. They morphologically resemble chlamydomonas. They morphologically resemble the chlamydomonas. They are ovate in shape. Broader posterior end and narrower apical end is present. Broader posterior end and narrow apical end. Narrow apical end and broader posterior end is present. They possess apically inserted equal flagella. They have eye spot, contractile vacuole and cup-shaped chloroplast with pyrenoid. Cup-shaped chloroplast with pyrenoid is present. Cup-shaped chloroplast with pyrenoid, and they are uninucleate structures. Cells are interconnected via cytoplasmic strands. Cells are interconnected via cytoplasmic strands, and posterior cells are typically larger than anterior cells. Clear? Everything is fine? Yeah. Asexual reproduction, which is very important. Just look into the process. Asexual reproduction occurs in some specialized cells called as gonidia. See, these are the specialized structures. I will explain here and later you can go through the theory. So, here gonidium, which is the specialized structure, here the asexual reproduction occurs. This undergoes sequential mitotic divisions, forms into 4-celled stage, later 8-celled stage is formed like this. 8-celled stage. 8-celled stage is known as Plakia stage. 8-celled stage is the Plakia stage, and this further develops into 16-celled stage, and here you can see small hollow like structure in this entire cavity. This is known as pilophore. From this hollow cavity inversion takes place. Okay, from this hollow cavity over here inversion takes place, where the entire colony is inverted like this. Entire colony is inverted, and you can see a complete inverted colony here. Complete inverted colony with pilophore, with filiophore inverted down, the cavity inverted down, and this gives rise to a complete young colony. A young colony is observed over here. Clear? Gonidium, specialized cell, followed by sequential mitotic division. At 8-celled condition, you will call it as Plachia stage, and this further undergoes mitotic division forming 16 celled states and a narrow cavity over here. You will address it as Filiophore. Then the inversion of colony takes place. Inversion of the colony takes place. Complete inverted colony you can see with a narrow cavity at the base, which is Filiophore, and this gives a complete young colony. This produces a complete young colony. Clear? Now, during sexual reproduction, in sexual reproduction, development of antheridia and development of oogonia is also observed. So, this complete theory once we will recall here in the asexual reproduction, then we will move on to sexual reproduction, gametogenesis and germination. Here you see asexual reproduction. This occurs by what? Gonidia. Specialized cells we have seen here. These cells are present in the posterior end. Okay. So, what are the steps here? Gonidium will withdraw the flagella and they will enlarge. They will divide mitotically and they form a 8-celled stage called as Plachia stage. Further divisions will give 16 plus cells which are arranged into a hollow sphere with a Filiophore. This hollow sphere is having what now? Filiophore, which is a small opening. The sphere will then undergo an inversion. The cells will develop both flagella and they form a new colony over here, which may or may not leave the parent. Next is what? Sexual reproduction. Sexual reproduction. This takes place only during unfavorable conditions. Sexual reproduction takes place during unfavorable condition. Here it is oogamous type of sexual reproduction. Fine. Cenobial type we can see both monoecious and dioecious types. Monoecious or bisexual, where from a single or same colony you can see both male and female daughter cells forming. Example of monoecious species, monoecious or bisexual, or you can simply call it as heterothalic. Monoecious or heterothalic. Okay. Volvax, globator. Both male and female sex organs are seen in the same colony. Protandrous condition is observed to prevent autogamy. Okay, where male organs will mature prior to the female sex organs. Dioecious condition, that is homothalic condition. Sorry, monoecious, bisexual or homothalic, where both the sex organs are seen in the same thallus. This is monoecious, bisexual or homothalic. Dioecious, unisexual or heterothalic.
Okay.
Example you can see Volvox aureus. Sex organs are present in different colonies. Fine, right? And then gametogenesis. Gametogenesis. Female gametes here are egg. Egg is the female gamete which develops in the posterior half of the cenobium. Okay, female gamete develops in the posterior half of the cenobium. This develops in posterior half of the cenobium, where they lose flagella and they become flask shaped, denser and darker than gonidia. Specialized structures than gonidia. They will become flask, dense shaped and darker structures. Next is male gametes or anthrozoids. They are from antheridium. They are formed via successive divisions. They result in 16 to 150 to 16 to 12 biflagellate anthrozoids. Anthrozoids are formed. Okay, male gametes or anthrozoids, they result in 16 to 12 biflagellate anthrozoids. The development is similar to that of zoospores, passing through Plachia stage, inversion and Filiophore. We have observed all these stages just now. Okay, in the explanation, this is the theory. Next is fertilization and zygote formation. How they are fertilized? Anthrozoids will be liberated. They will swim all across the water currents and they will move to the egg cells which are in the other colony. The anthrozoids fusing with the egg cell, they will form zygote or oosphere. zygote or oospore is formed. Oospore will secrete a 3-layered structure around the zygote. A 3-layered structure is being secreted which will protect the zygote, and this accumulates a hematochrome which gives orange red color. This is released by rupture of the colony, and this acts as the perinating body. Okay, this acts as perinating body. Now, how the germination process is done? Asexual reproduction we have seen from gonidium to the formation of young colony, and sexual reproduction also we have observed. Same the condition, the antheridium will be developing into a structure, a cavity, and spermatozoids will be released. They will swim across the water currents to reach the female cell. Here the development of oogonium, you can see how the antherozoids are entering the egg cell, and after which the zygote is formed. Germination of the oospore undergoes meiosis. Oospore will undergo meiosis. This type of meiosis is known as what? Zygotic meiosis. Oospore will undergo meiosis. What is the division here? Meiotic division, which is addressed as zygotic meiosis. They form 4 nuclei, 3 degenerate and 1 will survive. The survived nucleus will undergo mitosis. Single nucleus out of the 4 daughter cell, out of 4 daughter they will zygotic meiosis means all daughter cells will be haploid again. The haploid daughter cells, of which only 1 will be surviving, and that 1 will develop into a new cenobium. Clear, right? This is the general characteristics of chlorophyceae with clear type studies of chlorella and volvox. In our next class, we will be looking into rhodophyceae, phaeophyceae, its general characteristics, type studies. Lastly, we will also address cyanophycean members and economic importance of algae. We will finish the chapter of phycology. Then we will move on to mycology. Clear, right? So, that ends this session, and in our next class, we will meet up with phycology, and in our next class, we will meet up with phaeophyceae, rhodophyceae and cyanophyceae.